So with that, welcome everyone. We'll dive straight in. And Brene and Jessalyn, I'll hand over to you guys.
Sounds great. Brene and I are both from Duke University. I will go ahead and introduce myself, and then Brene will introduce herself, and then I'm going to give a bit of background on our lab and what motivated the study, and then Brene is going to dive into the details. So we're going to try to keep this short from the beginning. So I'm Jessalyn Dunn. I'm an assistant professor at Duke in biomedical engineering and biostatistics and bioinformatics. Our group works mainly in biomedical data science in trying to translate the use of large data generating platforms to making clinical decisions. And so we fit nicely between, I guess, the hardware and the software and the clinical decision-making components. My lab is the Big Ideas Lab that actually stands for something which I think annoys a lot of people, these acronyms, but it's the Biomedical Informatics Group, and we integrate data engineering and analytics. And really that describes what we do well, because we cannot only work with data in a silo, we need to bring the data together with the technologies that generate that data in order to make the most use of them. So with that, at the end of this session, hopefully, if we have a couple extra minutes, I'd love to talk about the COVIDentify study that we've launched recently to tackle COVID-19. But we're here today to talk about the work that Brene Bent led, who's my PhD student, and so I'm going to let her take it away.
Great, thank you. Hi, I'm Brene Bent. I am a PhD student in biomedical engineering at Duke University, working in the Big Ideas Lab. I'm in my 4th year, going into my 5th year. And I'll turn it back over to Jessalyn to talk about kind of our motivation behind doing this study, and then I'll jump into some more of the study details.
Great, thanks, Brene. So what really motivated this study was that we have done a lot of work with consumer wearable devices. So that would be devices coming from companies like Apple and Fitbit, Samsung, and so on. And our goal is to develop digital biomarkers from the data that we collect from these devices. And one of the big challenges that we were running into is that we couldn't find good evidence out there about the accuracy of the devices against clinical gold standards. And namely, one of the big concerns out there, especially in pop science literature, is that these devices may not work as well on people with various skin tones. So there's evidence to suggest that the green light, that's the PPG sensor that's used in the majority of consumer wearables, gets absorbed by darker skin tones more dramatically and might cause issues with signal to noise ratio and issues with accuracy of heart rate measurements. And so we wanted to first dive in and see if that issue exists, and if so, how bad is it? And we also wanted to understand, under varying activities of daily living, are there circumstances where the devices work better than other circumstances, and even are there devices that work better than other devices? So that's sort of what motivated us to do this study. And I think Brene can do a very good job, because she led the entire study of describing the setup of the study and what it is that we found.
Jen, do you think we could pop ahead a couple slides? Wonderful.
Absolutely, just tell me what you need. This one or the next one?
This one's perfect, and I'll just ask for the next one in just a minute. Thank you so much. So in this study, we were looking at 53 people with an approximately equal distribution of skin tones on the Fitzpatrick skin tone scale across 4 activity conditions. So we looked at folks when they were seated at rest, when they were walking, deep breathing, and performing a typing task. We examined errors in heart rate and heart rate variability, which is a metric of autonomic nervous system function. And we performed this experiment with 6 different wearable devices. These devices included 2 research-grade devices, the Empatica E4 and the BioVotion Avirion, as well as 4 consumer-grade devices that are commonly used for research studies, such as the Apple Watch, Fitbit, Garmin, and the Xiaomi Band, which is popular in China. These devices were validated against the clinical standard for heart rate, electrocardiogram, or EKG. In this study, we used a 3-lead EKG called the Bidium Pharos 180. Like Jessie Lynn said, we explored a variety of suspected inaccuracies in the optical heart rate measurement, including skin tone, motion artifacts across different activities, missingness, signal crossover, and signal lag. We examined error between the true heart rate measured by the EKG and the heart rate measured by each wearable device. Our findings, oh, can we skip ahead one slide, Jen? Thank you so much. So our findings showed large differences between the devices themselves, specifically significant differences between research grade and consumer grade devices. Interestingly, we found that research grade devices tended to be less accurate than consumer grade wearables. We also explored differences in error among those 4 different activity conditions. We saw an average accuracy decrease of 30% when a person was walking compared to when they were seated at rest. We also saw large errors during the typing task. These larger errors we saw in the walking and the typing, they could be indicative of signal crossover, which is when the device locks onto a repetitive signal, such as walking or jogging, or maybe in this case typing, and the device thinks that it's the cardiovascular cycle. We also did numerous analyses on different skin tones across the devices and found no significant differences of device accuracy between the different skin tone categories. We examined skin tones across the activities and found no significant differences in error in any of the activities or when combined across the skin tones. We also examined how much data was missing during the activities across skin tones, and again found no differences across the skin tones. We also looked at heart rate variability across the skin tone categories and again found no significant differences. Our statistical analyses included paired and unpaired t-tests with Welch approximation and Bonferroni multiple hypothesis correction, and we also performed mixed effects models with Bonferroni multiple hypothesis correction. So this was a very brief overview of all of the extensive analyses we performed in the study, and I hope we can jump into some more details in our discussion. Our main takeaway for you today, though, is that researchers, clinicians, and health consumers must recognize that information derived from these different wearables shouldn't be weighted equally for drawing study conclusions or combining study results or making health-related decisions. Algorithms that are used to calculate digital biomarkers should consider error and measurement quality under these various circumstances that we've shown here in this study. And digital biomarker interpretation must take this data quality into account when making healthcare decisions. So we're really excited, looking forward to your questions.
This is terrific. I'm going to switch across to our attendee list and remind folks either type your questions into the Q&A or please feel free to raise your hand and then I'll unmute you and we'll have you in direct conversation. While folks scramble to find their participant list in the Zoom, I wonder if I can kick off by asking you guys, I have 2 questions and take them in the order that's best for you. The first is, it's really interesting to actually see a very systematic comparison across consumer wearables and those other technologies that you describe as research grade wearables. So my 2 questions are, can you tell me, because I'm always learning about the right language, when you say a research grade wearable, does that mean sort of a cleared medical device, or is that a nuance that I'm missing? And then I wonder if you could dive in a little bit more about sort of the differences you found between those technologies. Oftentimes I've seen a device clearance being used as a surrogate measure of quality for use in research, for example, and I'm curious to hear your thoughts around that. So tackle those 2 in any order you like.
Sure. So I can just briefly talk about the nomenclature, and then maybe Brene wants to tackle the second question about their use. But ideally, what we're thinking about here is how we talk about fit for purpose, right? And I think in the V3 paper, we get to that quite a bit, which is another paper that we have coming out next week. But the idea is, when are these typically used, right? So for a lot of the fitness grade devices, we see that, or the consumer grade devices, we see that people are using them in their typical fitness routines or just in daily living, ideally not for making any sort of medical decisions, but just for kind of understanding the trajectory of their fitness level and health in some very broad strokes. With the research grade devices, these are the devices that we see being used in laboratories, that are being used to run studies in the field, almost as proxies for what we would typically see measured in a clinic. And so I guess what we're trying to get to here is the difference in how these are being used and whether they're fit for the purposes that the company advertises them to be used for.
Yeah, Jessica touched on the key difference in this study that between research grade and consumer wearables, which is that what are they marketed for? So the Empatica E4 and the BioVotion Avirion are marketed more towards the research and clinical research areas. And the consumer wearables, so Apple Watch, Fitbit, Garmin, Xiaomi Band, are devices that are marketed towards the consumer for consumer personal use. However, when we did a literature review of these wearables in research and clinical studies, we actually found that the consumer wearables were being used quite frequently, more frequently actually at the time of the publication than the research grade wearables, at least in terms of names popping up in these research and clinical studies. So that is one key difference, is what they're marketed for. And the data that you get from these devices is also very different. So if you look at the consumer grade wearables, we have a particular sampling rate. This is a dynamic sampling rate, but typically we're getting a heart rate measurement once every 5-ish seconds, and that varied greatly depending on the wearable and depending on the activity, which we can dive into if anybody's curious about the missingness aspect. So these devices have this dynamic changing, and they also provide us only heart rate. So they've already done all of the post-processing on the optical heart wave signal, and they pulled out the heart rate values with their own proprietary algorithms that we don't have access to, and may or may not have been validated. And that is compared to the research grade wearables, where they give us all of the raw data. So we have a much higher resolution data than we get for the consumer wearables. The research grade wearables also give us an algorithm derived heart rate, which was what was used for the majority of this study. So they give us a heart rate calculated with their algorithms, but this is also typically at a higher resolution. The Empatica, for example, has a 1 Hertz resolution for their heart rate. So once every second, you're getting a heart rate measurement. You have that dynamic sampling rate and you have the unknown sampling rate with those research grade devices, but you have a much higher sampling rate, which is very useful when you're looking at more sensitive metrics like heart rate variability. So I mentioned previously that we looked at heart rate variability across skin tones, but we were only able to do this with those research grade devices because we don't have a high enough resolution data with the consumer wearables to do that same analysis. Did that answer your question, Jen?
It absolutely did. I flicked across my screens to see if I was on mute before I started talking to you. Yeah, that's really helpful. And I wonder if I can pick up on something that you mentioned there. And definitely, I want to involve folks in the line on this too, because I can see a few names of people who I know have been doing this work too. You mentioned that you did a literature review, right, of the different ways that these tools were used before you dove in. And you also made a really good point, I think, in your opening remarks, Brene, around the importance of sort of really understanding the quality of the data that's coming off these tools before you plug them in as input data to sort of algorithms. One of the things that strikes me, and I'm being very selfish here because I'm thinking about, is this something that DIME needs to tackle? One of the things that strikes me is I don't see a standard approach to reporting this data and these performance characteristics in the literature. I wonder if you can comment on what you found during your literature review and what good might look like in terms of a sort of standard reporting structure for folks who don't have access to experts like you or labs like yours, and they want to know from other people's work instead of having to go back to right to the beginning whether a tool could work for them.
So this is a great question. This actually brings us to another project that we've been working on, which is called the Digital Biomarker Discovery Pipeline, or the DBDP. And this is an open source resource for people developing digital biomarkers that kind of goes through the process of pre-processing this data, trying to discover what devices might work for you, what softwares might work for you. And so that's something that we've been developing. And if you go to dbdp.org, you can find more information about that, and we're continually updating that. And this could be something that would be a really great addition to that. Actually, just kind of thinking about it is, what device might be the best use case for me? Right now, we're kind of expecting somebody to already have a device in mind, but it might be good to have this kind of guide from the beginning of what devices might be the best for my use case. And typically the devices that we saw used in this literature review were devices that they either had on hand or devices that maybe are a little bit easier to acquire or less expensive. So devices like the Fitbit was the most commonly used device across the studies that we saw. And this might be partly because of the relative inexpensiveness of the Fitbit compared to some of the research grade wearables, compared to perhaps something like the Apple Watch or some of the higher end Garmin devices. I don't know if Jessie Lynn has anything to add here.
Yeah, I think that's great. That's definitely, one of the projects that we're hoping will help to clarify this. But I think to your point, Jen, it is a big challenge because right now in the literature, there is not really consensus on what it takes to decide on whether a wearable is accurate enough or is at high resolution enough, or whatever your criteria is for the purpose that it's being used for. And so I think there are a lot of initiatives going on now to try to address that. I think the DIME Society and the Electra Labs libraries are two of those that are showing how these are being used, in what cases have they been used in the literature, and what are sort of the metrics that are being reported. And that's really helpful. What we found was that the difference between what's being reported about consumer wearables and what's being reported in scientific literature about non-commercial devices is kind of different. And so I actually, I think Brene may have a slide in here on the different metrics that we use to actually assess accuracy in this study. I think we have the NAE slide. Yeah, essentially, I think this is a good one here, where we're looking at sort of the mean absolute error and the mean directional error. And, what we were looking for initially in this project, which made this project, I think, even more of a challenge than we expected it to be, was, well, how are we going to decide whether or not these devices are good enough? What does good enough mean? And what are the metrics that we would use to evaluate these versus other devices potentially? And so Brene did a very deep dive into the literature on comparing different devices and how we could do this, given that the gold standard was not an equivalent measurement method. So it wasn't like we could really use necessarily the Bland-Altman method in its typical form. And so we use these two metrics, which I think are not typically used, but again, this isn't a typical study yet. We hope it becomes one. And I'm really curious from the group, actually, the feedback on these definitions of how we decide on metrics of accuracy and error, whether this is intuitive and makes sense, or whether there are other metrics that the group would maybe recommend looking into.
We have a question in from Michelle, but I'm going to shut up for a change for a second and allow other experts on the line to unmute themselves and respond to Jesse's question around sort of the approach to evaluation here. We'll let people noodle on that for a little bit longer. I want to circle back to it though, because I think, you know, pressure testing these approaches is really important as we try and hone in on a broadly accepted approach, right? If this doesn't feel right to any end user, we need to collectively sort of look at it again. I know the work that went into sort of establishing this, and we've certainly built on it in other work that we have to come out together, but I want to hear from the group. Michelle, you typed, Michelle Holko, we've got a couple on the line. You typed a really good question into the chat. Can you, are you somewhere where you can unmute yourself?
I can. You might hear my kids in the background, but.
The course, yeah. Michelle.
Happy to. So I just I wondered if you could dig in a little bit in terms of how you handle curation, especially across different devices when sampling frequency varies so dramatically. And also the types of data and outputs and sensors. And I don't know how much you've dug into the actual sensors and the engineering behind that and how much that impacts what you're getting out. And then kind of a related question is when you're doing these types of studies, do you create relationships with the vendors of the commercial devices so that you can get more raw signal, or do you just go with what the Fitbit NPR already provides?
So I'll let Jessie Lynn take that second question, but I can dive into the first bit. And I think the first bit that you said there was, there's a lot there, right? Jen, do you mind going to the slide that has equations on it? It looks rather mathy.
This one or there's one more?
I think that, yeah, this one.
Yeah.
So it was very interesting when doing this study because we were comparing everything to an EKG. And an EKG has a very high sampling resolution. 1,000 hertz was what we used, but in some cases that can be even higher. So it has an incredibly high resolution, and you can get heart rate, multiple heart rate samples every single second. And like I said earlier, the research grade devices like the Empatica, they give us a heart rate measurement every single second. And the consumer grade devices gave us a dynamic or changing sampling rate, which was approximately-ish about once every 5 seconds. And so it was a real challenge to compare EKG heart rate then to each single device heart rate, especially because these were all different. So what we did was we matched them based on matched timestamps. And the way we were able to make sure that our timestamps were matched across all of our devices was in our time protocol. And so we followed along with the network time protocol, and there's a bunch of ISO numbers that are in the paper that you can check out in the methods. But basically, we synced everything to an internet clock that was like our gold standard time, every single participant. So every participant, we synced up their time. And so the EKG had the exact same timestamp as each of the devices. And likewise, all the devices had the same exact time as themselves for each individual participant. And then for each participant, what we did then was we matched a timestamp given by the EKG with the timestamp given by that device. And so we had in total a number of matched timestamps, and this differed by device and by participant. So typically this study ran for about 45 minutes, but sometimes it took a little bit longer to run. Sometimes we were really efficient. It took a little bit shorter to run. So you got a different time, but then also a different number of matched timestamps depending on the sampling rate of that device. So we were able to match up that timestamp with the EKG timestamp. And that's where you get the top of these equations here, which is the heart rate EKG minus the wearable heart rate. And we summed all of these up and divided that by the number of total matched timestamps to get a mean directional error for each participant. We also, if you look at the second equation, we did mean absolute error, and where we just took the absolute value of the EKG minus the wearable and got rid of the sign differences, because sometimes it sampled above the EKG or the heart rate was above the EKG heart rate, and then sometimes it sampled below and it was below the EKG heart rate. With these equations, then we were able to determine error between the EKG and the wearable. And we did this calculation for every participant in our study, 53 participants, and for each one of the devices. So there were 6 devices. So we did each of these calculations for all of those different conditions. And then I will pass the torch on to Jessie Lynn to talk about your second question, which was talking about the devices.
Yeah, so I think this was the question about the relationships with companies and whether we tend to work with them. So I would say for this particular study, we actually didn't work with any of the companies. And, you know, I think in the end, this was an ideal setup because we were trying to be as objective as scientifically possible by not having any funding or any external companies involved with the study. The reason for that is that we wanted to see how these devices would be used in an actual study. So our goal was to mimic as best as possible what would go on if a researcher purchases a device, which is what we did. None of these devices were donated. We purchased the devices. And we ran the study in our own lab, and we compared it directly to a different device that we also purchased. And so what we're hoping at this point, and a lot of companies have actually reached out to us since the study came out, which I think is maybe not surprising because, you know, I think companies' goals are to do right by researchers and to make sure that, you know, they're at least trying to figure out what it is that needs to be done. And I think one of the challenges before was that it wasn't really known what needs to be done to make sure that these devices can be used for digital biomarker development purposes. And so this was kind of a first pass, and I think there's a lot more to do here. But the fact that companies, I think particularly Empatica, has been very interested in working with us, and a couple other companies have also reached out that want to make sure that they're incorporating the framework that we've built here into their own studies for verifying and validating their devices.
Thank you. That was all very helpful.
And Jesselyn and Brene, those were terrific answers, and I took a few notes while you were speaking. I also, Michelle, sort of related to your question and related back to the study. I had a conversation that I think folks might be interested in with a few industry colleagues earlier this week, and they were asking very similar questions, Michelle, around, you know, we're starting to see that performance of commercial technologies can actually be quite good, and they may be fit for purpose today for our study needs. What makes them nervous is the risk of, say, in 6 months' time, there being a difference in the way that the data is sampled, or the difference in the way it's made available to the sponsor, and does that lend it to being an appropriate sort of research tool? We also took a sidebar conversation, and it's timely, and I found it fascinating, so I'm going to bore you all with it now, with, you know, this idea of compatibility and the different ways that we sample, not being new to the era of remote connected sensors. And we were actually talking about ventilators, which is very timely. And the way that a ventilator in a hospital, for example, depending on the company, might report the number of oxygen coming from a patient during their respiration. There's one big company that I won't name that does, I think, oxygen sampling every 5 seconds, and another one that does it per breath. And what that's meant is that, you know, large hospital systems sometimes buy data integrators to be able to feed that information into the EHR. Other smaller hospitals or healthcare systems are actually basically bound to a single technology. And I think that I'm curious to hear from folks on the line and also from Jessie Lynn and Brene about, you know, how can we learn from our colleagues in this sort of imaging world and the medical devices world as we move really quickly in these sort of connected sensors to try and make implementation as easy as possible. And the silence is the problem that I'm having, because I feel like this is something that we need to tackle, you know, and whether it's, you know, is it sort of pooling together the research community to say, these are our needs, right? These, if you're a commercial technology or otherwise, this is what we need in terms of access to more unprocessed data or particular sampling frequencies, or the agreement to hold firm on a given protocol for a given period of time. And those are things we're kicking around, and certainly either on the call now or afterwards, I'd really value some feedback on that. And Michelle, it sounds like you're thinking similarly in terms of the deployment of these tools based on your question.
Yeah, absolutely.
Yeah, I think, you know, one of the things that I think an idea that we've kicked around quite a bit is also this idea of companies pushing software updates and not making those updates public. And so I think that kind of what you're saying about these changes in sampling rates and the changes even in the formatting of the data and the changes in the methods to collect the data, all of that is not well documented and not publicized or even made public. And so I think that's one of the big challenges from the research standpoint is, if the data that we're working with today is different than the data that we're working with yesterday, we need to know that, because we make a lot of assumptions when we run statistical tests about the data that we're dealing with. And one of the big challenges here is this breakdown in communication between what's going on the device technology side and what's going on the data analysis side. And I think it's the responsibility of everyone in that pipeline to bridge that gap, to make sure that any assumptions that are being made in the data analysis phase are appropriate for the data that we're using. And the only way we can know that is to be in the loop with these companies that are pushing software updates, changing backend technologies on a regular basis. So having some framework in place for keeping those changes organized and having a method of communicating those regularly to data analysts, I think is really what we need. I don't know quite what that would look like yet, but at least from our standpoint, a lot of times we are making a lot of assumptions and hoping for the best because there's not much we can do otherwise. But more information, always useful.
Fantastic. Christine, I'm going to unmute you if I can do that successfully. Yeah, go ahead, I can hear you.
I was just thinking about that and just had like a comment to build on and maybe a question from there. So I'm at Electra and we're building, you know, the evidence library for all these different tools. So we have the Charge 2, the Charge 3, the Charge HR, and now the Charge 4, which is coming out this month. And it's really hard for us to make recommendations to our pharma clients because the Charge 4 might be what they can buy and buy in bulk and distribute, but all the evidence exists for the Charge 2 and the Charge 3. Not even the Charge 3. That one like doesn't have a whole lot yet. So it's hard to make those like real-time recommendations when you're not sure if the evidence for the Charge 2 still even applies or is valid. So it's almost like wasted work if you can't like make that connection for people.
Christine, can I riff off that question and ask Jesselyn and Brene, when, and I'm going to link it back to our V3 paper eventually because I'm shameless like that. When, can you help me understand, so if in the example that Christine gave, which makes sense for a commercial project, product, new versions, right? Then people want to go out and buy, there's a reason to buy. It makes total sense from a business standpoint. what do you expect to see and what do you actually see in terms of documentation around what the changes are, and trying to use that information to map back to Christine's question about how can you know whether previous bodies of evidence still holds?
Yeah, this is a great question, Jen and Christine. It was extremely challenging to find any information from these device companies on their validation procedure, on how they went about standardizing anything across different software versions. It was difficult to find which software version was on the device itself, let alone any documentation for that. Companies are understandably not open at all about their validation process. And if these devices are going to be used in the research community, in the clinical community, we really need these device manufacturers to be more open about how they're validating these devices. Because right now there is hardly any information out there. The research grade devices do a little bit better job of partnering with folks in the academic community to work on validation processes and procedures for their devices, but in consumer grade wearables, I don't know of any documentation that exists, at least among the devices that we've studied in this particular study.
That's wild, to think that it doesn't exist. And again, you go back to what's the target market for the commercial products, and it's consumers using it for health and wellness. So it's hard to be critical, but it puts your question into focus, Christine, doesn't it? Which is, if you can't access the information, and again, Jesse, that goes back to your comment about sometimes not even knowing when these updates and things have been shipped, let alone sort of understanding their implications. That unknown, there's a very limited tolerance for that when you're trying to use these tools to answer very specific scientific questions, and especially in particular across regulated studies where you really need to have that documentation closely on hand. I'm going to hold. Oh, go ahead.
No, I just, this is Michelle again. I just wanted to, knock on that because, that's something that we're starting to encounter with the All of Us research program too, is especially with Fitbit, you can certainly get information about the device easily, but knowing what software or what version of algorithms they're running is a challenge. So it's going to be interesting to maybe even start to do some discovery on our end of can we see where those changes happen just based on the patterns of the data. And I wonder if anybody has done something like that, Jessie Lynn.
Yeah, we did exactly that actually with a device that we used previously. The Basis watch that ended up getting bought by Intel before it sort of got kind of shut down. We had seen in our data some very strange trends where everybody's heart rate seemed to increase all in one day, and it didn't seem like it made a whole lot of sense. And so luckily at that time, the company was pretty open. And I think it's actually a big shame that Basis is no longer here and that a lot of these companies that were once startups are now these huge companies, because when they were more in the startup phase, they were a lot more open about what they were doing. And so I actually had a conversation with the folks at Basis and said, we're seeing some weird things in our data. And they were like, oh yeah, we pushed a software update that day. Here's what the software update did. And we saw it in our data and we said, that makes absolute sense. And now when we're building our statistical models, we're building a different model for pre-software update and post-software update. And after that, we asked them, we said, can you send us the schedule of the updates that you push that we would expect to have seen made a difference on our end? And they did that. I would say at this point in time, I can't imagine that a company like Fitbit or Apple would be willing to give that sort of information, which is really unfortunate because it really throws a wrench in a lot of the modeling that we're trying to do. So I guess to your question, can we try to sort of reverse engineer from the data and detect when software updates were made? If you have enough data and you're not doing an intervention study, then yes. So having controls is really important. But again, there's a lot of things that are difficult to control for, like seasonality, like just external influences, like the COVID crisis right now, right? Like you'll definitely see changes in smartwatch data because you have these large scale events happening. So I think a lot of what I like to push here is kind of an injection of domain knowledge, to think really hard when you see changes and trends in your data about where that could have come from. And also just showing it to a lot of people. I think a lot of times one of the benefits of we have a super diverse lab, people come from all sorts of backgrounds, and some of the ideas that people have when we put a graph up on the screen are something that I would have never thought of, maybe Brene would have never thought of. And it's just the fact that you have these diverse backgrounds coming to the table helps you to brainstorm all of the possible things that could be going on.
Yeah,
that's great. Thank you.
I'd like to jump off of that with a couple of points. The first point is, if you have a study with a wearable device and you haven't started the study yet, put a pause on all software updates for the study. That is a huge recommendation. We did that for this study because they actually did push software updates in the middle of this study, but we had at the beginning set all of those software updates to not go into effect, and so we were using the same version throughout the study. So if you're doing a study where you're able to do that, I highly recommend that. And then the second point is something that kind of jumps off of what Jen had mentioned a little bit earlier. And earlier this week, I was giving a talk on this same study, and I got a really interesting question. And that was, are we going to try to validate the EKG that's on the new Apple Watches? And the EKG on the Apple Watches has gone through the FDA regulatory process. So I explained to the person who asked the question that it's already been validated in an open process, that we as the public can have some access to this validation process. And I think something needs to happen with these wearable devices if they're going to be used in research, and especially if they're going to be used in the clinic to make actionable health insights. We really do need to have some regulatory process of validating these devices and validating the software. And I think Jen and Jessie Lynn and our co-authors on the V3 paper would definitely agree with that. There needs to be some process for validating the data from these devices, because right now there is nothing for at least the optical heart rate sensor on these devices.
So Brene, I'm going to riff off that for a second, and then I'm going to hand back to you guys with the last question, and then we'll close out. I know that everyone's back to back today, and I want to make sure that folks have an opportunity to grab a turkey sandwich or something before your next call. So we've mentioned a couple of times the V3 paper. So for folks on the line who might not know, Brene, Jesselyn, actually Christine on the line as well, and a broad range of other experts from DIME, and this harkens back to Jessie's really good comment about the need for an interdisciplinary look at this, work together to think about what fit for purpose means when you're evaluating a biometric monitoring technology, and came up with a sort of three-step process: verification of the sensor, analytical validation of the data processing, and then clinical validation as we actually infer clinical meaning from all of that. Part of that work includes sort of reflecting on the different triggers that might lead you to need to repeat some of those processes. So things like shipping software updates, things like expanding the context of use perhaps to new patient populations. So that is due to come out on Tuesday. So keep your eyes peeled for that. And sort of a lot of the brilliant work that happens in Jesselyn and Brene's lab helped inform our thinking on that. So really appreciate you guys. Before we move on to close out, I do want to take just a quick pause. Jessie and Brene, do you want to talk about your COVID Identify study? Because I think it's something that a lot of people on the line will be interested in and may even have some questions on.
Yeah, absolutely. And thanks for giving us a chance to talk about it, because it's been all-consuming for the last probably about month. Essentially what we're doing here is we are trying to build upon a study that we completed a while back, where we've been working on digital biomarkers of infectious disease and flu. And when I say digital biomarkers, what I'm talking about here are using commercial wearables and the signatures that we can measure from those to detect flu or infection, which we've done pretty successfully in the past. And with the COVID-19 crisis coming online, it just seemed kind of like a very reasonable extension of what we've already done. And at the same time, a huge amount of data that would help us to really refine the algorithms that we've built to potentially get more specific to COVID, to better understand how some people tend to seem to kind of have very mild symptoms or get better really quickly, whereas other people tend to kind of crash, and it's really unclear why there's this big difference. I mean, in some cases it's obvious, and in some cases it's totally unexpected. And we could potentially use wearables to help understand that process. And then the third piece that we're really thinking about is resource allocation, that if we are able to predict even a few days in advance where hotspots are coming online, where ventilators may be needed, we can really help, you know, even like the North Carolina government has reached out to us to try to figure out how can we do a better job at tackling this and making sure that we're early, so we're not faced with a situation where we have an inundation of patients. So the study itself, if you want to take a look, it's at covidentify.org. So that's COVIDENTIFY. I think, Cove Identify.
I think you got it.
Hopefully I've spelled that correctly. I'm not a spelling bee champ or anything, so. But if you go there, you can read more about it. If you're interested, you can sign up. And essentially the way that it works is the study emails 2 questions a day. We had the email of the option of both email and text message, and one of the things that blew up this week was that the carrier started flagging our text messages as spam. So we're working on that, but for now it's via email. So you can sign up via email and get those 2 text messages per day. They ask, were you within 10 feet of anybody outside of your household, and do you feel sick? And if you answer yes to feeling sick, we ask a few follow-up questions. If you have a Fitbit or Garmin, you can link that now to our study and we can actually start pulling in that data. If you have an Apple Watch or any other wearable that links to Apple Health Kit, we are building an app that should be ready in about 2 weeks that can pull in all the rest of that data. And so what we really hope is that we can get ahead of the curves, at least, you know, it's tough because we are not used to working at this kind of pace, as I think Jen knows and is quite frustrated by. Academic science moves at a snail's pace, usually. So, you know, it's been a lot of staying up, pulling all-nighters for the last month, just trying to make this study happen. And so we're hoping we can make a dent somehow in the crisis.
And Jessie, can I just ask you to share, because I've been really impressed, a lot of the questions around using these connected sensor technologies for sort of public health surveillance is around the privacy and the sort of data rights of the individual. How are you tackling that with the Covidentify study? Because I think you've done some nice work here.
Yeah, and that is one of the most common questions that we get. And so one of the reasons it took us so long to get the study off the ground was actually our IRB here at Duke is very, they have good oversight, very good oversight, which is great, I think, because it forces us to think about the hard questions. And one of the things that we've been really careful about is, okay, what is the data that we're pulling in? Where does that data go? Is it possible to re-identify participants from that data? And if so, because in a lot of cases with studies, that is inherent to a study, that if somebody's giving their name and phone number, you know, you can figure out who that person is, right? And so what we try to do is we have a lot built into our back end to separate out the identifiers from the rest of the data. But, you know, there are some arguments to be made that if we have somebody who maybe has a palsy or some very characteristic way of moving, that even from movement data and zip code, you could potentially figure out who that person is, right? And, you know, these are very difficult questions to answer. A lot of these studies haven't yet been done to figure out what it means to re-identify somebody just from, for example, their heart rate and zip code. Is that possible? But we're trying to get ahead of that problem by doing our best to separate out the data. So our wearables data sits in one database on the Duke Protected Network. We have a study identifier that's the only identifying piece of information about that wearables data, and that's a map. So basically, I'm explaining a lot of the details of our backend, which maybe is too much detail for you, but I know the privacy folks care about detail. And so I guess the take-home message is when we have identifiers, we only give that identifying information to the study team members who need that information to do their jobs. So like my study coordinator, for example, needs to be able to call participants directly and reach out to them. And she has to be able to say, hi, Mr. or Mrs. Jones, you know, I'm calling from this study. So it would be impossible for us to not collect identifying information. The way that we use it, we are very careful about.
Fantastic. I appreciate you taking a bit of a dive into that. And I think sort of separating out the data pools and even getting into the details is helpful as we try and make everyone more comfortable with what sort of, you know, best practices are to be able to work with other collaborators and sort of insist on seeing that. So I really appreciate you getting into that, Jessie. So I want to make sure we leave on time. Brene, Jesselyn, this was terrific. Congratulations on a great study. Congratulations on getting COVID-Identify off the ground and sort of leading the way with that work. I appreciate everyone joining on the line today. It's also really nice just to get people together. I know it's a tough time sort of for everyone right now. So I particularly enjoy these touch points. Before we break, I'm just going to highlight a couple of upcoming Dine activities that folks might be interested in. I'm really excited. At the end of this month, we have our next lightning speed crowdsourcing session, and as many students from grad school all the way down to undergrad are having a very different experience of their sort of spring semesters. We're devoting it to great ideas and work done by our students. And I think, Brene, you have set the bar incredibly high for what brilliant PhD candidates can do in this field. So this will be a great discussion where the next generation of leaders in our field will be seeking feedback from our community. And then our next journal club is going to be Abhi Pratap, and he's going to be looking at a huge study that was published about a month ago, thinking about specifically retention and how retention is different across different subpopulations. There's some terrific work, and Abhi will be here discussing that with us next month. And our next webinar is actually on digital therapeutics. So we talk a lot about digital medicine. Digital therapeutics are an important part of digital medicine. So we have Megan Coder from the Digital Therapeutics Alliance and Jeff Abraham from Achille coming in to talk to us about digital therapeutics and give us a shared functional knowledge of what we mean when we use that term. So again, Brene, Jesselyn, thank you. I know you are both so busy. I appreciate you joining us and chatting with us today. Everyone on the line, thanks for spending your lunchtime or your morning coffee with us today. It was great to connect. Really appreciate this tremendous work, and stay safe, everyone.